Hello friends, in this video we will discuss about the anatomy of monocot stem. In last video we shall discuss about the anatomy of dicot stem. You know, these two steps, dicot and monocots, is the most important difference between these two stems because in dicot stem these vascular tissues are present or well developed, and it is a secondary growth is present in the dicot stem of monocot plants. But in monocot stem these vascular tissues are not well developed, means the secondary growth is absent in the monocot plants. This is the main difference between the dicot and monocots. The dicots, they are present the conjoined, collateral and open type of the vascular bundles, but in monocots having the conjoined, collateral and closed type of the vascular bundles. Means the monocot plants, this cambium get absent, and that's why the secondary growth is absent. Now in this video we will discuss about the anatomy of monocot stem. The tears or transverse section of monocot stem showing different layers. These first one important parts, it is a epidermis. It is a single layer structure, and this epidermis in monocot stem, they are not having any interruption or root hairs are absent in the stem of monocots, and also the root, these trichomes are absent in the monocot stem. These trichomes, hair-like outgrowth, is absent in the monocot stem, and this trichome is also present in the dicot stem, but in the monocot these trichomes are absent in the epidermis region. It is a single layered structure. Next one part is below this epidermis present the hyphodermis, and this hyphodermis having the 2 to 3 layers, and this hypodermis cells, they are sclerenchymatous tissues, means the sclerenchyma is found in this region of the monocot stem. And below internal structure or central parts of this monocot stem, they having the xylem and phloems tissues, but cambium is absent in the monocot stem. Now the structure of this stem, this tears or transverse section of this stem showing different layers. This outermost layer, it is called epidermis. Simple structure of this monocot stem, outermost part, it is a epidermis. Below this epidermis present the hyphodermis. Below the epidermis present hyphodermis, and central parts present the vascular bundles, having the vascular bundles. Also present the metaxylems and protoxylems. This tissue and also phloem is present in this region. These are the vascular bundles, and inner parts, this is the vascular bundles. And the central parts, it is called ground tissues. This is a simple structure or tears of monocot stem. This showing the outermost layer, it is the epidermis. Below the epidermis having hyphodermis, and it is a ground tissues, and these ground tissues, some scatter cells are present, and these scatter cells are the vascular bundles. They are found in the innermost part of the monocot stem. This hypodermis, it is a sclerenchymatous. Below this hypodermis, they are found the central parts like the ground tissues, and these ground tissues means these ground tissues, in this ground tissues there are interruption, means the present the vascular tissues in these ground tissues, means they are scattered. And these vascular bundles are scatteredly present in these ground tissues and having the xylem and phloems. In monocot stem, these metaxylems and protoxylems arises at this position, and outer surface they can surrounding the phloems tissue in the monocot stem. Means these vascular bundles is the numerous, and these vascular bundles they are scattered in the ground tissue. This epidermis they do not have the cuticles, means the cuticle is absent. Below this cuticle the hypodermis, and this hypodermis having the sclerenchymatous cells or sclerenchymatous tissues, and this sclerenchyma it is a hard bast, means it is a dead tissues are found in this region of the hypodermis, and the vascular bundles they are numerous, and these vascular bundles they are scattered in the ground tissues. And important points, this lysigenous cavity is also found in the vascular bundles is in the xylem is a endarch condition of the xylem. Xylem is endarch. Xylem is endarch condition, and this xylem they can present sclerenchymatous bundles with cells, and these vascular bundles they can showing the lysigenous cavity. This lysigenous cavity is formed because this cavity means the protoxylem cells they are disintegrate and they can form the this lysigenous cavity is formed in the xylems, and is most important characteristics of this monocot stem. This cambium is absent because the secondary growth is absent in the monocot plants, and the secondary growth means this cambium is absent, only the phloem and xylem. These tissues they are found in the stem of monocot plants, and most important pith is a central location in dicot stem, but pith is absent in the monocots. This pith is a central core in dicot stem, but in the monocot this pith is absent. It is the most important characters of the monocot stem. Now in this video we will discuss the anatomy of monocot stem, and this anatomy means it is a differ from the dicot stem, because in the monocots their secondary growth is totally absent, because this cambium layer is absent, means cambium is absent in between the xylem and phloem, but in dicot stem it is present, and that's why in the dicot plant secondary growth is also present. And these several layers are shown in the monocots, because these monocots vascular bundles of this monocot means they are scattered position in the ground tissue. This is the anatomy of monocot stem.